So my lab generally focuses on developing genetic tools for non-model microbes, and the human microbiome is a great means to put that into practice, just because the vast majority of microbes you find in those communities have no genetic tools in place. And so that's what is the major umbrella that my work focuses on. And what I was talking about as part of the conference is my work developing a new generation of antimicrobials, ones that can be programmed to go after some microbes but not others. And the goal is that by developing those, you can go into these complex microbial communities, such as in the gut, on the skin, other locations, and eliminate individual members, and in turn study how the microbiome responds to that, and also potentially as a therapeutic if you want to eliminate the pathogens while preserving all the beneficial bacteria that are present. So synthetic biology is a field focused on engineering biology, much like you would a car, an airplane, a computer, and has really been advancing by leaps and bounds over the past decade with all these wonderful advances in new technologies and capabilities. And I was actually a graduate student in one of the first synthetic biology labs. So that's where my start was, in synthetic biology. And it was only in the past 4 years that I made this transition into the microbiome. It was a really unique opportunity to apply the skills and techniques and overall principles that my group has been developing, but in this case to a really important problem, which is being able to understand and eventually engineer the human microbiome. All right, so CRISPR is now widely known as a revolutionary genome editing tool. It's a very exciting technology, but also makes for an extremely competitive field. I was fortunate that I got my start in it when CRISPR was still a quirky immune system in bacteria, and before it was known as this very wonderful, powerful tool. And so it allowed me to establish some foundation, but it's still an extremely competitive field that I do my best to stay on top of. Right, so there are many bottlenecks that are certainly in place. If I hope to go from where I am now, developing really a platform technology, into something that's in the hand of clinicians or in the general public. Right now we're working on the bottleneck of delivery and figuring out how to get CRISPR into a given set of cells and having it faithfully expressed and kill off the cells we want to get rid of and not harm the other cells. Oh, for me, it's having to jump into new disciplines I've never been familiar with. So my background was in synthetic biology and RNA engineering, and it was transitioning from that to learning about CRISPR and how to engineer it. And in moving into microbiomes, it's learning about the microbiome field, and now in trying to tackle delivery, learning how to engineer bacteriophages. And so having to pick up these new fields has been a challenge, but also admittedly one of the funnest, most rewarding parts of the job. So in my mind, microbiome engineering is making small, defined changes to a microbial community. It could be adding in new members, or eliminating what's already there, or just trying to tweak the composition. And that stands in contrast to FMT, where you're making a large bulk change. You're literally replacing one community with another. And so the hope is we better understand the contributions of the individual members of the community and how we can affect the overall community by changing those members. Eventually we can make very small defined changes that still have these very large beneficial effects. So the whole basis of my work is using CRISPR to selectively target sequences present only in some microbes but not others. And the idea is when CRISPR recognizes that sequence, cleaves the genome, kills the bacterium. And if that sequence is absent, the bacterium is spared. And so by picking sequences present only in the target bacteria, we can go after the things we want to eliminate and in the process spare everything else. We're getting there. Really focusing on this CRISPR antimicrobial as a platform technology, but in the back of our minds are thinking through what the most viable paths forward are, not only in terms of technical feasibility, but also from a regulatory and even a commercial standpoint. So excited to see what the future holds as we continue moving forward. So we plan to continue forward. Thankfully, we have a good amount of funding in place and are beginning to branch out as we look not only developing the platform technology, but applying it to potential human pathogens as well as plant pathogens, and even thinking about it in the context of contamination in industrial processes. So the hope that there could be lots of different applications that emerge from this one project.